If you happen to be walking out in the arid open plains of southwest Queensland, Australia, you might see the black head of a snake appearing out of a crack in the dry ground, basking in the intense heat of the Australian sun. While it might not look as immediately threatening as a cobra or a rattlesnake, this is an animal you want to avoid at all costs. It's the inland taipan, the most venomous snake in the world. According to its lethality rating, the LD50, its venom is about 7 times more deadly than that of the hook-nosed sea snake, around 23 times more potent than the Indian cobra, and 72 times more deadly than the venom of the king cobra. One bite from the king cobra releases, on average, 420 milligrams of venom, enough to kill around 2,600 mice. But one bite from the inland taipan, which only releases about 44 milligrams of venom on average, could kill up to 220,000 mice. And in some cases, inland taipans have been shown to inject over 110 milligrams of venom in a single bite. That's enough to kill 500,000 mice, or over 100 humans. Even a small bite from an inland taipan can cause permanent damage, and if you're not given the taipan antivenom within 45 minutes, you'll most likely bleed out from your wound, your kidneys will shut down, and your body will go into paralysis, leading to respiratory failure and death. Before antivenom was readily available, every bite from an inland taipan was fatal. But why is it so lethal? And how does it do so much damage to the human body? And how did it evolve to become the most dangerous venom in the world? To understand why the inland taipan is so deadly, we first need to understand where venom emerged in the evolutionary tree. Like so many hunting and feeding traits in animals, venom in snakes appeared due to dramatic changes in geography and habitat over millions of years. It's not clear exactly when and how venom appeared in snakes. It could have either come from a single origin around 170 million years ago, leading to toxicity in the venom of many diverse reptile species, or it could have evolved independently across multiple lineages. So we might not know exactly when it evolved, but it's generally understood why it evolved. Based on fossil records, it's been suggested that the first snakes caught and killed their prey by mechanical constriction. Like modern constrictors, these early snakes had robust vertebrae and short but powerful muscles between the vertebrae, allowing the spine to flex at multiple positions. A snake constricts its prey by capturing it within several loops in the spine, and then applying pressure, using the short distances between the vertebrae to create tight angles within the loops. The prey's blood flow to vital organs, the brain and the heart, is restricted, causing the animal to fall unconscious and eventually die from cardiac arrest. The forest-rich habitats of the Jurassic era were perfectly suited to this exact sort of predation. A snake could hide in the dense undergrowth, patiently waiting for its prey to pass, grabbing it quickly and trapping it between the loops of its spine before it could escape. The relationship between a snake's method of killing their prey and their habitat is something we still observe today. We tend to find constricting snakes like boas in jungles and dense environments, as these habitats are perfect for ambushing their prey. But what happens if you open up the environment? During the Miocene period, from about 23 to 6 million years ago, environments across the world began to slowly change. The air became cooler, the conditions drier, and differences in local climates became more pronounced, leading to areas opening up to form savannas. These were perfect for the development of rodent and bird life, but less ideal for the constricting snakes. The snakes that were ideally suited for constriction weren't suited to chasing animals across these new, more open landscapes. This required longer, multi-segmented muscle chains instead of shorter, more powerful blocks of muscles used for constriction. And so, if snakes needed to chase their prey, they also needed a new way to immobilize and kill them: venom. Over millions of years of evolution, snake venom has developed into a complex cocktail of biological toxins that can stop prey dead in their tracks. In most cases, the amount and toxicity of the venom delivered by a bite is far beyond what is required to kill a single animal. But why does it need to be so lethal? It's because venom serves many different functions. It prevents prey from escaping, it calms them down, stopping a return attack, and it also, very importantly, begins the process of digestion. Killing the prey is an added bonus, not just because it means that they can't run away, but also because it speeds up the process of extracting the necessary nutrients from the animal. But what's really astonishing about snake venom is not only has it evolved to be highly toxic to a snake's target prey, venom has also found more than one way of achieving this toxicity. The black mamba, native to Africa, and the coastal taipan, native to Australia, are both members of the elapid genus. They both have similar body sizes, color, and venom toxicity, and they both hunt similar-sized mammals like rats and mice. But the biological components of their venom is very different. The black mamba's venom is mostly made up from so-called Kunitz-type peptides and three-finger alpha neurotoxins. But the coastal taipan venom is made up of mostly phospholipases, beta neurotoxins which have a completely different mechanism of action to alpha neurotoxins. We'll get on to how these different toxins work later, but this is just one example of convergent evolution. The venom of these two species are designed to do exactly the same thing, but have a very different composition. We see these large and small variations in venom composition in similar snakes across the world. Let's take the three Australasian taipans as another example. The Central Ranges taipan, found in the mountainous regions of Western Australia, the coastal taipan, found along the east coast of Australia and the southern border of Papua New Guinea, and of course the inland taipan, found in the outback of Western Queensland. Each species survives on an entirely mammalian diet, and they all eat similar-sized animals, but there are surprising variations in the composition of their venoms. The Central Ranges taipan, like the black mamba, has venom made up from almost entirely three-finger alpha neurotoxins. Coastal taipan venom, as we saw before, is mostly made up from phospholipase beta neurotoxins. And the inland taipan has a deadly combination of both of these types of neurotoxins. It's difficult to say exactly why the inland taipan has evolved such a potent venom. It might be because of the scarcity and difficulty of catching its prey in the open areas of the outback. But it could also be as a result of random gene mutations that haven't been selected against. Either way, there's no denying its toxicity. Despite how powerful its venom is, the inland taipan will bite its prey multiple times, recoiling after each strike to allow the venom to surge through the body. It injects a mixture of toxic proteins into the bloodstream and subcutaneous fat, which begin to act instantly. First, one type of enzyme breaks down the proteins in the blood and the blood vessels, loosening the connective tissue, weakening the walls of the capillaries, and triggering inflammation in the body. Alongside this, another type of enzyme helps to increase the rate of absorption and spread of the venom, acting as a sort of venom accelerant. So, what actually kills the animal? Well, first there are the hemotoxins. Inland taipan venom contains a number of proteins which encourage clotting of the blood. On small amounts of blood outside the body, this can cause it to turn into jelly, causing almost complete coagulation. But when acting on much larger volumes of blood inside the body, the opposite occurs. Thousands of small clots are formed very rapidly, using up almost all of the blood clotting factors. So instead of causing coagulation of the blood, it actually prevents blood from clotting at the site of the injury, leading to potentially lethal external bleeding. The next organs that are vulnerable are the kidneys. These might be directly damaged by certain proteins in the inland taipan venom, or what's more likely, indirectly damaged by toxins that affect the muscles. These myotoxins specifically target muscle fibers, causing the release of damaged muscle tissue into the bloodstream. The kidneys then kick into action, trying to remove the damaged tissue from the blood, but the myoglobin in the muscle fibers is toxic to the kidneys, blocking the kidney's complex filtration system and causing the organs to fail. On top of this, the now severely damaged and almost dead skeletal muscle pulls large amounts of fluid from the blood, sending the body into shock. But if the animal, by some miracle, manages to survive the bleeding and kidney failure caused by this first set of toxic proteins, there is almost no chance it will survive the effects of the second set, the neurotoxins, the proteins that are primarily responsible for the venom's remarkable lethality. As we saw with black mamba and coastal taipan venom, these toxins are separated into two categories: alpha and beta neurotoxins. The alpha neurotoxins in inland taipan venom are fast-acting, deadly proteins, targeting acetylcholine receptors at the junction between the muscles and the nervous system. Acetylcholine is a vital neurotransmitter which allows the nervous system to communicate with the muscles, controlling the contraction and relaxation of muscle fibers. So when the venom's alpha neurotoxins bind to these receptors, they prevent acetylcholine from binding, stopping communication between the brain and the muscles, leading to rapid muscular paralysis. But despite how dangerous this sounds, this process is relatively easy to reverse. The binding of the toxin to the receptor isn't very strong, and if you're bitten, the paralysis caused by these toxins can be overridden by administering an antivenom, as long as it's injected within 30 minutes of the bite. But things aren't quite as simple for the other type of neurotoxin in inland taipan venom, the beta neurotoxins. These are much slower acting, but considerably more lethal. The primary beta neurotoxin in inland taipan venom is called paradoxin, and is part of a family of enzymatic proteins that are found naturally within cells. But in most cells, their activity and concentration are carefully controlled due to the dramatic effect they can have on the cellular machinery. Paradoxin primarily acts on the components of the cell membrane, specifically targeting the chemical bond which joins the long-chain fatty acid to the glycerol molecule within a phospholipid. Paradoxin breaks this bond, which, by itself, is enough to cause significant damage to a cell, potentially rupturing the cell membrane. But it's actually the product of this reaction that causes the biggest problems. When the phospholipid is broken, it releases a fatty acid chain, specifically a molecule called arachidonic acid, and it's this that ultimately leads to permanent paralysis and death. Again, like with the alpha neurotoxins, the site of action of paradoxin is the neuromuscular junction, the point of connection between the nervous system and the muscles. If paradoxin gets into the presynaptic cell, it'll start by breaking down phospholipids to produce arachidonic acid, which then triggers a stream of calcium ions to be released. Cells are very sensitive to changes in the amount of certain ions, and this large increase in calcium ion concentration causes almost all of the acetylcholine to be released from the synapse. The acetylcholine travels across the synapse and binds to its receptors, causing them to become desensitized and turn off for a short period of time. By itself, this wouldn't be enough to cause paralysis, but now there's a critical problem with the communication system. Almost all of the acetylcholine has now been used up, and even if the cell wanted to make more, it can't, because the arachidonic acid has also prevented reuptake of choline, the molecule required to make acetylcholine. But this isn't the only problem caused by paradoxin. If it gets into the post-synapse, it can also deactivate acetylcholine receptors completely. Again, the same thing happens. Phospholipids are broken down into arachidonic acid, which causes a massive release of calcium ions. But this time, the calcium ions activate enzymes which turn off the acetylcholine receptors, even causing them to be removed from the cell membrane for a number of hours. The result of all of this is a complete and irreversible shutdown of the neuromuscular junction. The brain can't consciously or unconsciously communicate with the muscles, and the body is totally paralyzed. The lungs can't inflate, the heart stops beating, and the animal dies of respiratory failure. There are many types of these beta neurotoxins found in different snake venoms, but paradoxin is the most potent of them all. This by itself would make the venom lethal, but when combined with the many other toxic proteins that affect multiple systems and organs in the body, it makes inland taipan venom not only the most dangerous snake venom in the world, but also one of the most dangerous naturally occurring toxins ever to be discovered. The natural world can be equally devastating and astonishing. While it's fair if you don't want to marvel at the wonder of snake venom up close, or even come within 1,000 feet of an inland taipan, it's hard to deny that they are incredible creatures. The best appreciation for the natural world comes from understanding it, and understanding it comes from grasping concepts across many subjects, from chemistry to biology to math to physics. And since it's been a while since my high school and college science classes, I like to brush up on these concepts, to engage my brain, and to help continuously fulfill my curiosity for the natural world. And to do this, I use Brilliant. Brilliant is an amazing tool for learning STEM interactively. It's less like a college textbook and more like a series of quizzes, puzzles, and funny animations that make the learning engaging. Oftentimes, the courses feel like a game or riddle that leave me feeling satisfied when I get the answer right. But if you get stuck on a particular question, Brilliant doesn't penalize you or impede your progress. Instead, they give you an in-depth explanation to guide you to the right one, so you can learn from your mistakes. There are so many courses to dive into, from algorithm fundamentals to logic and probability to mathematical fundamentals, which is the course I'm currently working on. This course covers the foundational ideas of algebra, number theory, and logic that come up in nearly every topic across STEM. To get started for free, visit brilliant.org/realscience or click on the link in the description, and the first 200 people will get 20% off Brilliant's annual premium subscription. And if you're looking for something else to watch right now, you can watch our previous video about the spy pigeons of the Cold War. Or watch Real Engineering's latest video about the way engineering solved the mystery of the Concorde crash.